Now we announce the webinar begins. Let's welcome the Chair Dr. Childers' address.
Bella, thank you. Very well done. And I'd like to say that even though this is a new journal, it is an opportunity for those of us in the field to get papers published. And I'm very happy to report that so far we've received some very good quality, primarily review papers on DMD. And so we look forward to getting this special edition into press as soon as possible. We still have opportunities to receive additional papers. All right, so that's enough. So let me introduce David. David Mack is a colleague, a close friend, and associate professor at the University of Washington. David's trained as a geneticist, and he's really been in the field for quite some time now. And he's really focused on modeling, human induced pluripotent stem cells for neuromuscular disease. So David, I'll let you take it away, and you've got 20 minutes beginning now.
All right, thanks, Casey. I'm trying to share, but I'm not succeeding. What do you guys see?
Yeah, we can see your slides.
Yeah, and if you try to go to the presentation mode, let's see. There we go. Now we're seeing your version. Maybe if you switch.
Yeah.
Yeah, switch display. Perfect. We're good.
All right. Yeah, thanks, Casey, and thanks for including me in this kickoff beta testing for RDODJ. The story I want to present is based on the premise that measuring clinically relevant outcome measures like contractile force is a good way to test for new drugs and to look for disease etiology for neuromuscular diseases, and that measuring the restoration of that function is a powerful way to validate new therapeutics. So the data I'll show today is based on our modeling of the cardiomyopathy associated with Duchenne muscular dystrophy and the skeletal muscle defects. But just keep in mind that the platform that I'll be presenting can be applied to any neuromuscular disease. So here are my disclosures. And for those of you who don't think about this so-called disease in a dish idea, I want to take you through the workflow that we commonly use in my laboratory. So the idea is that a child with Duchenne muscular dystrophy comes into the clinic. We can collect their urine, and in that urine are sloughed kidney nephron cells that we can then expand in culture to capture that patient's genotype. Then we use standard reprogramming factors to turn those urine-derived cells to iPSCs. And then the trick is to figure out the in vitro differentiation scheme that recapitulates embryonic development so we can turn those iPS cells into the tissues that are diseased in the patient. So we make cardiomyocytes to model the cardiomyopathy and skeletal muscle to model the skeletal muscle defects. We spend most of our time in the lower right quadrant, which is trying to identify the molecular drivers of the pathology, and can we identify the disease phenotype that matches the presentation of the patients. And given our track record, as Casey mentioned, given our track record in AAV-mediated gene transfer, we use that phenotype that we've identified. We can give gene transfer, we can do CRISPR gene editing to ask, can we reverse the phenotype in the dish? And if we can, then we take that drug candidate into a normal drug validation platform with the idea of then delivering that back to the patient that donated the urine sample. So we are all very familiar with the etiology of Duchenne muscular dystrophy and the phenotypic manifestations that we know in young boys. So I omitted that from this presentation, but I want to reorient you toward, for the rest of the talk, shift your attention to the role of dystrophin in embryonic myogenesis. So the work I'll be presenting today is part of a larger study between my lab and the lab of Lisa Maves at Seattle Children's Research Institute. And the idea is to look at DMD from a developmental perspective using 3 models to uncover the evolutionarily conserved molecular drivers of muscle pathology. So the immediate goals are to identify the earliest transcriptomic and epigenetic perturbations downstream of dystrophin deficiency during embryonic myogenesis and uncover epigenetic modulators capable of restoring muscle function in all 3 models. So today I won't talk about any of the epigenetic dysregulation, but I will end the talk with some transcriptomic data that we've generated recently. So let's first talk about the cardiomyopathy. In DMD, the cardiomyopathy is caused by inflammation, cycles of cardiomyocyte death, and fibrosis. With the initial fibrosis, patients present with diastolic dysfunction, as well as ECG abnormalities and arrhythmias. As the fibrosis continues to accumulate, patients present with systolic dysfunction and reduced left ventricular ejection fraction and cardiac remodeling, and ultimately the disease progresses to the point of end-stage heart failure. While 25% of patients present with signs of cardiomyopathy at age 6, most are not treated until they reach their teens. So let's break down the pathological progression of the cardiomyopathy. Without dystrophin, starting at the top, the DGC does not properly localize to the sarcolemma, which leads to membrane instability, dysregulation of calcium carrying, phosphorylation of the ryanodine receptor, elevated resting cytosolic calcium, and impaired calcium reuptake. The increased calcium causes increased calcium or reactive oxygen species and mitochondrial damage. The dysregulation of nitric oxide signaling further exacerbates this aberrant calcium handling and mitochondrial dysfunction. And all of these diverse mechanisms coalesce into the death of cardiomyocytes via the calcium-dependent protease-mediated degradation and engagement of the mitochondrial death pathway. So it's the cycle of dystrophin deficiency and this cardiomyocyte death, deposition of fibrosis, and this cycle continues until we see heart failure. So to remind you where we've been, we spent several years studying the DMD cardiomyopathy in 2D. So we turn iPS cells into cardiomyocytes on flat plastic. And to summarize 10 years of work in one slide, we were able to show that starting in the lower left, that we see increases in membrane fragility, increases in release of injury markers like CKMB. We also see increased intracellular calcium, increased spontaneous electrical activity, and a decrease in calcium transient kinetics like calcium reuptake. We also see defects in the mitochondria, reactive oxygen species, and mitochondrial damage. And in the lower right, that all culminates in a decrease in myofibril force, force kinetics, calcium sensitivity, and we see some impact on cardiomyocyte and sarcomere alignment. So we then, even though I've just shown you, we can recapitulate multiple aspects of the disease phenotype in 2D, we thought it was really important to try to model the cardiomyopathy in 3D, and I'll show you why in a minute. So a very talented bioengineering graduate student in the lab, Samantha Bremner, took on the project to create these so-called engineered heart tissues. So what you're looking at on the left are 12 cardiomyocyte, iPS-derived cardiomyocytes in an engineered heart tissue, and hopefully you can see them contracting between these two posts. And I'll explain more about that in a minute. So the idea is that these EHTs can generate force, and looking at that force output and the kinetics of contraction and relaxation, we can get further insight into the disease etiology. So this is what the representative images from normal and dystrophic EHTs. They show differences in sarcomere organization as shown in the F-actin staining. Although we do see alignment in the direction of uniaxial tension, the DMD EHTs show shorter sarcomere length, but no difference in Z-disc width. So in the absence of dystrophin, you can see here on the lower right, let me get my laser pointer on. You can see an absence of dystrophin in this EHT. So before I show you the data of the contractile force and kinetics, you need to understand the platform that we've helped our colleagues at Curie Bio develop over the last 8 years or so. The platform we've developed is called MANTA-Ray, which stands for Magnetometric Analyzer for Engineered Tissue Array. The idea is that these engineered heart tissues are suspended between two posts, one rigid and one flexible. There's an embedded magnet in the flexible post to measure the voltage change as the magnetic flux moves in space, and there's a GMR sensor under or outside the well. And by knowing the stiffness of the post and the amount it deflects, we can measure the force output using this simple equation. So this is the manta ray instrument shown on the right. It's about the size of a shoebox and it fits in the incubator. What I'll show you the data from today is a 24 well version, and we also have a 96 well version under development. So manta ray has an intuitive user interface to control stimulation parameters. I can start the movie. So what's shown is each well can be controlled individually. The white borders tell you which wells are being monitored. The two left wells are low current stim with increasing left to right, and absolute force is tracked on the y-axis. So you can change the voltage, you can change the current, the pulse duration, pulse polarity, and the pulse frequency. To give you an idea of the kinds of tests that you can do in this platform, we can do treated versus untreated, you can do dose response curves, you can do target engagement by adding the drug and then doing a washout. I'll show you examples of twitch and tetanus ratios. We can do force frequency curves, and we can measure fatigue over time. So for this brief talk, just keep in mind that we can learn a lot about the muscle biology just by looking at the contractile kinetics. So what the various parameters that are shown on these curves, I don't have time to unpack all of these today, but I'll show you examples of each one of these domains. So these data have recently been published, so I'll go through it really quickly. With an N of about 30 per genotype, Samantha was able to show that the DMD EHTs produce less force under oxytonic conditions, meaning the muscle is shortening under increasing load. So focusing on the red line in the upper left panel, you can see the normalized force tracing showing the slower onset of contraction and a pronounced delay in relaxation. One of the main messages of my talk is that we highly value this functional outcome measure of force, so this accurate and reproducible assessment of contractile force was one of our main goals of developing this platform. So in some of the data I'll show you, the tracings are shown on the left and the quantitation of that data is shown on the right. So we can measure twitch force, twitch stress, active twitch power, and twitch work. If you then normalize each one of these curves so they can be overlaid, you can then also see a delay of contraction onset and a pronounced delay of relaxation. And those are also quantitated on the right. We can measure calcium transients using the ratiometric dye Fura2. In data I don't have time to talk about today, we also showed that putting the EHTs under load improves these calcium transient kinetics, making the measurements more physiological. So now focusing on the red tracing in panel A, the calcium flux measurements revealed higher resting and peak calcium in the dystrophic EHTs, but a decreased amplitude of the calcium flux. And again, quantitated on the right. So we see higher baseline calcium, higher peak calcium, but a lower calcium amplitude. Again, standardizing those curves so they can be overlaid, we see a delayed release of calcium into the sarcolemma and a delayed calcium reuptake. So the time to peak is slower, the time to 50% decay is faster, but the rate of the overall decay is also faster. So I don't have time to go over this in great detail, but the main point I want to convey with this transcriptomic study that we did is that making engineered heart tissues matters. And what I mean by that, if you look at the transcriptomic changes that occur in iPS-derived cardiomyocytes, dystrophic cardiomyocytes in 2D, the story that prevails, it's all over the place. We can't make sense of the various parameters that are changing between the normal and the dystrophic cardiomyocytes. However, when we turn these cardiomyocytes into engineered heart tissues, we see physiological relevance that's directly related to the absence of dystrophin. So if you look at the upregulated genes in these dystrophic EHTs, you see the changes in regulation of membrane potential, changes in heart contraction, and changes in cardiac muscle tissue development. And on the downregulated transcripts, we see calcium ion homeostasis disrupted and extracellular matrix organization disrupted. And again, we can talk about more in the discussion, but in these engineered heart tissues, we saw physiologically relevant transcriptomic changes. Okay, for the last few minutes, I want to now shift to the skeletal muscle defect in DMD, and I can tell you that making skeletal muscle from iPS cells is much harder than making cardiomyocytes. So the idea is that we push these iPS cells into mesoderm, and then by modulating Wnt and BMP, we push them away from lateral plate mesoderm into paraxial mesoderm, because that's the source of axial and limb muscles. What's shown on the bottom is the differentiation scheme to go from iPS cells to myoblasts. It takes approximately 28 days. And for the aficionados in the audience, these are the small molecule inductive and inhibitory factors that we use to drive this differentiation. But for the purpose of this short talk, just appreciate that we can make aligned hypertrophic contractile myofibers with highly organized sarcomeres. Okay, so much like we did before, we're able to make skeletal muscle in the dish. Once we turn these iPS cells into myoblasts, we then seed them into troughs and take them through secondary myogenesis, through serum starvation and insulin signaling, which takes an additional 10 to 14 days, and we can turn these myoblasts into really nice myotubes. So what's shown here on the bottom under 1 Hertz broad field electrical stimulation, these muscle tissues contract upon stimulation. So a research assistant professor in the lab, Alex Smith, was able to make engineered muscle tissues, or EMTs, with in the absence of dystrophin. And these EMTs produce about half the twitch force as compared to normal, and they produce about 1/10 the titanic force compared to normal. One thing to note here, that the twitch tetanus ratio that we're able to achieve in the dish is about 2 to 2.5 fold. As I'll show you in a minute, usually what we like to see a 10-fold difference between the twitch and tetanus ratio that we see when we do these in intact muscles in animals. So with about N of 12 per group, Alec was able to show a decrease in twitch force, decrease in titanic force, the contraction velocity is slower, the relaxation velocity is slower, the time to 50% and time to 90% relaxation are also slower. We did see a slight decrease in the DMD EMT cross-sectional area, but we don't think this is big enough to account for these substantial differences in force output. Just to give you an idea of how we're trying to relate these engineered muscle tissues measured in manta ray with physiological relevant measures in the animal, we're able to measure fatigue over time. So a 10 volt, 10 millisecond, 1 Hertz stimulation over 30 seconds, we can see that the DMD EMTs are more susceptible to fatigue. And just like I showed you in the engineered heart tissues, in these dystrophin knockout engineered skeletal muscle tissues, we see an absence of dystrophin here, this absence of the ringed fibers in these tissues. On the functional readout side, the last thing I want to present is that we're starting to ask whether the data we're collecting in manta ray can be correlated in any meaningful way with the types of data we've generated over the years in XLMTM and the DMD dogs. So what's shown on the right is the forelimb and hind limb force testing that we do in the dogs. You can see this nice torque frequency curve. What's shown on the left is a force frequency curve that we're able to generate in these engineered muscle tissues. The last thing I want to present is, in the last 2 minutes, is we've now done transcriptomic studies in these engineered muscle tissues over multiple stages of embryonic muscle development. So what's shown on the left is a T-SNE plot of iPSCs starting here. You can see they're nicely clustered. And as they march through the myoblast, the iPS to myoblast differentiation scheme over 28 days, you can see they cluster very nicely, although they do become a little more heterogeneous as they approach the myoblast phase. And what you can see is a nice progression through these mesoderm and skeletal muscle markers as they march through this 28 days of skeletal muscle differentiation. So this slide is one of the main take-home lessons of the talk. So now I've switched the axis and the iPSCs are shown at the top. This is a comparison of normal iPS-derived skeletal muscle compared with dystrophin-deficient iPS skeletal muscle. So as they march through this differentiation over 28 days, we know that in this in vitro system, that dystrophin is expressed at about day 7. So just downstream of an absence of dystrophin, you see this transcriptomic divergence. And by 28 days, we see 2 distinct clusters, normal and dystrophin deficient myoblasts. Shown a different way, the iPSCs are shown in the upper right, and as they march through this muscle differentiation, at day 7 to 10, we see this divergence where the normal wild-type iPSCs collect on one arm and the DMD, the skeletal muscle progenitors accumulate on the other arm. So we're seeing distinct transcriptomic changes that occurs over time, and you can track that in real time. We're collaborating with a company in France called Vidium, and they're able to turn this into a Google Earth-like clickable system. So each one of these dots can be clicked on, and it will show you the global transcript of each individual cell as it marches through embryonic muscle development. I think I'll skip this part because this is basically a rehashing of the same idea. So let's focus on this day 10 bifurcation point. A story that's starting to come into focus is that dystrophin deficiency leads to dysregulation of cell junction proteins during this somite formation. So at this day 10 bifurcation point, you can see the healthy myoblast progenitors on one side, the DMD myoblast progenitors on the other side. So, and the transcripts that are the most dysregulated at this bifurcation point are cell substrate adhesion, cell matrix adhesion, cell-cell adhesion, cell migration, and cell junction organization. And the 3 categories of cell junction proteins that are the most dysregulated are integrins, cadherins, and collagens. So this is starting to suggest that the earliest manifestation of dystrophin deficiency is a deleterious effect on compartmentalization and somite formation during embryonic muscle development. So we're postulating that a dystrophin mutation has this effect at this compartmentalization of the somite, shown here in blue, or during the delamination and migration of myogenic progenitors as they form the muscles of the body wall and limb buds. It could also cause a defect in this so-called epithelial to mesenchymal transition that occurs on the ventral side of the myotome, shown here, this kind of green and lime green. And the descendants of those myotome-forming cells are destined to become the pools of embryonic muscle cells that account for the huge expansion to create the muscle mass at birth and are the source of satellite cells. So stay tuned to see if we can really show that this is true, but our early data suggests that an abnormality in cell-cell junctions might be the earliest manifestation of a dystrophin deficiency. I'd like to thank the team who did the work and our funding sources, and I'd be glad to stick around and answer questions. Thanks.
David, thank you. That was a huge amount of data that you presented in 20 minutes flat, and fantastic. Really, really terrific. So thank you for that. Why don't we just pause for a minute to see if there are any questions from the audience? And then we'll move on next to Dr. Townsend. Questions, comments? So let me ask a question, David.
Sure.
The manta ray, the contractile apparatus that you demonstrated, that's commercially available, and can be acquired from academic laboratories and stuff like that?
Yeah, sure. Thanks for the prompt. So our long-term partners in this is Curie Bio. The manta ray system is commercially available. And what comes along with that is as much hand-holding as you need to get the system up and running. And that comes from some people that are now Curie Bio employees that were trained in my lab. So, yeah, it's an expensive. The capital outlay is pretty expensive, but we're still working on modifying the system to do the full panel of metrics for cardiac and skeletal muscle functional output measures. So if that's something you're interested in, please don't hesitate to contact me, and I'll put you in touch with the folks at Curie Bio who would be glad to help you get that set up.